This is our last seminar for the CBAC seminars, and we're very honored to have Professor Riccardo Olcese here from University of California in Los Angeles. He's a professor of anesthesiology and physiology, and recently he developed protocols to do a protocol known as voltage clamp fluorometry on the L-type calcium channel, and it's a story our lab's been following very closely. And we've been very impressed with the insight that he's gotten into the channel function and also its pharmacology. So hopefully we'll learn some of that today.
Thank you, Jonathan, for come here, and I guess it's green, yes. And yes, I had a wonderful day, and I would like to thank all the faculty, students, and postdoc that met with me today. And so today, I would like to tell you a bit about a new idea that we developed about antiarrhythmic drugs. And as Jonathan mentioned, the fluorometry project that we are right now doing at UCLA, well, it's not about that, the talk, unfortunately. So it's about something, let's say, more translational. My lab is split between the biophysical, ion channel, and a new idea that came from an interaction with the group of cardiologists at UCLA. And so it's about arrhythmia and maybe some idea about a new approach to suppress trigger of arrhythmia, like early after depolarization. So, yes, I shouldn't probably present this. We are all very familiar with this picture. Sudden cardiac death is like an epidemic in the United States. Yeah, no, the number varies between 350 and 500,000 deaths per year. So it's a significant problem. And you see, if that happened outside the hospital, the chance of survival is pretty much very slim. And even in hospital, you have a bit more chance to survive. But yes, as we know, it's an electrical problem that makes the heart going from a normal, well-organized electrical activity to a completely disordered electrical pattern. And yes, sudden cardiac death accounts for probably 50% at least of the cause of death in the United States. And yeah, I learned that it's more than all type of cancer together. So it's a significant problem, and as I said, it's an electrical problem of the heart. And we know that a normal event of excitability in the heart produces an electrocardiographic record that we recognize as normal here, and it's at the cellular level arises from the activity of a single myocyte, the cardiac action potential, right? And we are all familiar with this type of activity, of this type of excitation in the single cell, where the cardiac action potential is supposed to always monotonically repolarize down to the resting membrane potential. Now, this is a normal action potential. So under conditions that could be of various etiology, the normal repolarization is interrupted by this sudden oscillation. It's actually an oscillation of the membrane potential, right? As any oscillation, you have two fighters. That there must be a depolarizing current fighting a hyperpolarizing current, and you need to also add some fatigue. So the system starts oscillation. Fatigue could be, for example, the inactivation of one of the conductors. So these are very arrhythmogenic events, and they are called early after depolarization. Usually occur in phase 2 and 3 of the cardiac action potential. And so in the heart, a normal action potential is associated with a normal ECG, right? And when a single level, we observe this oscillation early after depolarization, these are the substrate for PVC, premature ventricular complex or contraction. These are extra beats that are particularly dangerous as they initiate a new depolarizing wave across the heart that can end up in wave break, VT, and ventricular tachycardia and ventricular fibrillation. So we are very good at pushing the heart or a single myocyte to go from a normal action potential, normal heart rhythm, into this pathological activity of early after depolarization in PVC. And these are caused usually by, we can use in the lab oxidative stress. Hypokalemia is also very good at triggering early after depolarization and PVC. But really the problem is how to go back from this situation to a normal heart excitability. So the talk today is about how can we restore and maybe explore some new idea. Well, this is what I was mentioning. EAD are dangerous because they propagate. And these are a nice work from my colleague at UCLA that show, for example, in a cable, and there is a few cells that display an early after depolarization. And you see 69 cells do not produce an extra beat or an extra action potential because they are clamped from the surrounding cell that do not want to have an early after depolarization. And you see how sharp it is? You just need one more cell. This source sinks mismatch produce an extra beat that propagates. And this is the way that we find the extra beat of the PVC, let's say, in the heart, in the tissue. So at the single cell level, this is a ventricular rabbit cardiomyocyte. We can paste an isolated cell in current clamp, expose the cell to hydrogen peroxide. This is a good way to simulate like an oxidative stress. And within minutes, maybe 3, 5, 6, 7 minutes, action potential, normal repolarization get interrupted by this oscillation early after depolarization, right? If we repeat the same type of experiment in a perfused heart under Langendorf, and we perfuse hydrogen peroxide, in this case was 600 micromolar. We found a good balance, 600 micromolar, to initiate and observe early after the depolarization within 5-10 minutes, while the experiment prolonged too much. So in the heart, we need less concentration. 100 micromolar is sufficient in the full heart. And you see, this is like a perfused heart. We are recording a pseudo ECG with bipolar electrode. And we also have a microelectrode inserted in the ventricle, sampling the voltage probably of a single cell or single myocyte. And you see, under this condition of hydrogen peroxide, you have early after depolarization, a couple of them in 2 beats. Then you have a run of ventricular tachycardia that transition in fibrillation. So we can have the same type of insult explore at the cellular level and in the full heart. So what's going on under early after depolarization? So as I said, they arise around phase 2 and 3 of the action potential. So we need to look quickly at the current flowing during a cardiac action potential, right? So I superimpose an action potential with early after depolarization. If we look right there where the whole thing starts, the only inward depolarizing self-regenerative current that is flowing in this moment in time, where irrelevant to EAD formation, is actually the L-type calcium current, right? There's something interesting. It's not the all L-type calcium current. It's not the peak. We have good reason to believe that this initial phase is very important for contraction to produce EC coupling and calcium-induced calcium release. So contractility probably is produced mainly by the calcium influx in the early stage, but there is like a late calcium current flowing toward the late phase of the action potential that seems very much involved in or correlated to the onset of early after depolarization. Why this current is important? Because as I said, for this early after depolarization to propagate, you need a self-regenerating current. So calcium come in, depolarize the membrane, like a positive feedback. So you can have this wave producing an extra bit. So we thought that if we want to suppress EAD-mediated arrhythmia, but we should maybe learn more about this late current and the properties, and the properties of late current, how they associate with early after depolarization. It is like an action potential clamp, and if we stimulate a myocyte with this waveform, we can actually see the nifedipine-sensitive current that is actually flowing or responsible for this upstroke in the middle of the action potential. So what is responsible for this current? Yeah, we know it's a macromolecular complex, the L-type CAV 1.2 channel. It's a monster, right? Made at least by 3 different subunits, a pore and regulatory subunit beta and alpha to delta. So this complex generates, under a constant depolarization, this nice fast inactivating current, right? And yes, there is an early peak that is the one that really necessary for effective contractility and calcium-induced calcium release. There is a late phase. You see, the current never inactivates completely, but you have like a remaining current there. So this sort of current flowing toward the end of an action potential and more, let's say, in quasi-steady state condition, right? So if we want to really look at the biophysical property of in steady state of this current, we look at these two curves, right? That really are the steady state curve that describe steady state activation and steady state inactivation. And the intersection of these two curves define a range of potential that are very important for the activation or reactivation of calcium channel that are ready to activate and not fully inactivate. So there is an interesting relation between this area and the steady state current that flow toward the end of the action potential of the late L-type calcium current. So you remember, you challenge the cell with hydrogen peroxide and EAD appears. You challenge the heart with hydrogen peroxide and transitioning first VT and then ventricular fibrillation. So what happened to the L-type calcium current directly when we insult a cell with hydrogen peroxide? So what we found A norm in control, nice action potential, hydrogen peroxide. You see a kinetic effect here is a bit slower, a bit more significant late current, and early after depolarization. Look at the IV curve. It's shifted a bit toward hyperpolarized potential. So there is an hyper excitability of the channel. When we plot the steady state curve, and remember, I like to point out about this window current region. Well, the activation curve is shifted a bit to the left. This is the overall effect of hydrogen peroxide on the nifedipine sensitive current. You have a shift to the left of the activation curve. The steady state activation curve is a bit to the right, and this non-inactivating component is also increased. So you have this width of current, which is a range of potential where the channel operates, which now is all of a sudden expanded, right? And so there must be a correlation between this magnification of window current region, the appearance of EAD, and arrhythmia. And yes, this is the picture. So, of course, you want to devise a therapeutic intervention, and you say, oh, well, you need a drug that push the activation curve to the right, the steady state activation curve to the left, and the pedestal back to like a few % of the total current. That would be, like, of course, a dream drug. So the question was, do we need to take care of all these three parameters to suppress EAD? Or there is some more sensitive aspect in that we can actually hit and suppress this oscillation, this event. And, well, I'm not the first one that look at the L-type window current region, and they're relevant to EAD. Here, Craig January and Riddle, 1989, and they were studying early after the polarization, and say our results suggest an essential role for L-type calcium window current and provide framework to understand the role of several membrane currents and the induction and block of EAD. So there was already, many years ago, this idea that the window current and the EAD were correlated. What is surprising to me is that never an antiarrhythmic strategy has been developed based on this idea. And, class 4 antiarrhythmics are calcium channel blocker, and they work, they suppress arrhythmia, except that they suppress contractility. So my colleague, cardiologist at UCLA, tell me they have very poor applicability in clinical practice because of their negative inotropy. So how to explore the role of this change in steady state activation and change in window current property, and how do they affect early after depolarization? Well, you should engineer channel and maybe an animal to have a different position of the activation GV curve, or different position of the steady state activation, or different pedestal. So if you can imagine, or maybe find drugs that are selective enough to be sure that they only touch one of them and sort out the mechanism, or at least the intervention that suppress this. So that would have taken probably several years. But there's another way, which is the dynamic clamp. I don't know if you are familiar with this, but the idea is very simple but very powerful. So you can run an experiment, a real experiment, with a myocyte in current clamp, in parallel with a mathematical model that run, for example, the L-type calcium current, and feed at any moment the membrane potential in real time to the model computes a conductance and gives back to the cell. Real time, like the myocyte doesn't even know it has a new conductance coming from a virtual channel, right? The good thing is since this virtual conduction is generated by a model, then you have parameters like steady state inactivation, steady state activation, the slope, the V-half, time constant. So you can play with this virtual current the way you want and understand what is the impact on the action potential or early after depolarization. So the idea is this one. You have a cell, you record in current clamp, you have an action potential, you feed this output to a model. The model is much more complicated than just a calcium channel. It's a full cell model of myocyte from which you output only the L-type calcium current model that has been fit on to the real experimental data. So you generate this current and then you give back to the cell. Now, you need to remove the endogenous L-type current, right? Otherwise you double two current together, which is very easy for the L-type channel because they're very sensitive to nifedipine. So you can do your experiment in nifedipine, and that's it. You feed back to the cell a virtual L-type calcium current. And see, and this is a typical experiment. We pace the myocyte, this is a rabbit ventricular myocyte, every 6 seconds usually. It's important, this bradycardia, to produce early after depolarization. We expose to 600 micromolar H2O2. EAD starts appearing. Then you block the L-type current with nifedipine. Of course, the action potential gets shorter. There is no more plateau. There is no more the L-type endogenous current flowing. And then we engage the dynamic clamp, and we put back a current with the property of the one that we saw modified by early after depolarization, by hydrogen peroxide, and EAD appear very consistently. You move the voltage dependency of activation, in this case by, I believe, 5 millivolts, EAD disappear. What I was amazed when we were doing this, when I was doing this experiment, is the sensitivity of the position of this curve on early depolarization. I could see, like even 1 millivolt shift, you could see already a decrease in the frequency of EAD. So we have something interesting here. And the beauty of this is you can move this curve back and forward, CAD, make them disappear. Like in the cell, you remember, it's still there, soaked with hydrogen peroxide. The insult is still there. You just shift this conductance voltage dependency by very little. And this is the quantification of this experiment. When you have the position of the GV curve where hydrogen peroxide shift the curve, you have an IPD 90 of more than 1 second. This is because every action potential have oscillation in EAD. And then you move the curve, in this case is 4 millivolts, in the direction where you are reducing the wind of current region. And look, the drop in the action potential duration and the drop in the frequency of EAD. Here, 90% of action potential display at least one oscillation. You move the curve 4 millivolts, you drop to 20%. If you do another millivolt, pretty much EAD disappears. So it's extreme sensitivity to this. This moving the activation curve in a way that reduces the overlapping, the wind of current region. So we did another experiment. Yes? This is a rabbit ventricular myocyte. Yeah, they beat. When you pace, they beat. Yes. Yeah. These are in current clamp mode. It's exactly a current clamp experiment, except that the calcium L-type nifedipine sensitive current, the conductor, is replaced by a computer-generated virtual current in real time. So, excuse me? 37 degrees. 37, no. is more like 34. This is our 37. We find very difficult to work. These are very long experiments, you can imagine. Yes, 34, 35. That's more or less the temperature And so now we can push the steady state inactivation curve in the opposite direction. Yeah, and the same. The sensitivity of EAD to this, the position of this curve is impressive. Like a few millivolts, you see the incidence or the frequency of early after depolarization dropping, dramatically. So you remember, so we are narrowing the wind of current, pushing activation to the right, inactivation to the left. There is another maneuver that you remember. This pedestal current is also very important. It's part of the wind of current, right? This is all the overlapping of the two curves and the range of potential where channel can operate and activate. So we also try to reduce this pedestal current, the non-inactivating component. The experiment is the same. We pace the myocytes at every 6 seconds, more or less. It's always a combination of bradycardia and some insult. It could be hypokalemia. It could be, in this case, hydrogen peroxide. And it's a very good intervention to produce early after depolarization in a reliable way. And we add hydrogen peroxide here. Again, action potential display early after depolarization within 5 minutes. We block the current with nifedipine, and we engage the dynamic clamp. We see the action potential getting prolonged again. Some small early after depolarization. While we are doing this experiment, we also discovered that to get really big swing in early after depolarization, you need to increase by a little, 10% IKS, right? IKS is a calcium sensitive, so probably since there is no more calcium coming into the cell, because now it's virtual, there is no real calcium coming, you need to boost a bit IKS, but given that, nice oscillation, very nice. And you have 80% of of action potential displaying early after depolarization, and of course action potential is very much prolonged because you have this oscillation going. You reduce the pedestal current back to normal, and from 10 to 4%, more or less, are the number we got from the single cell experiment of the L-type calcium current. Yes, EAD disappear, and the action potential become normal. So of course, this is like making a long story short. I show you the 3 best maneuver to suppress EAD. But in the meantime, we have explored many other parameters. The slope of this curve, we didn't know, are important. It's steep, slower. The slope of the steady state inactivation. The kinetics of the current. And this is an ugly figure, but it summarizes the effort we made in changing parameter of the slope, the kinetic of activation and inactivation of the channel. The only intervention that restore in a short, say, normal action potential with no EAD was the reduction of the pedestal current, the shift of the steady state inactivation in the direction that reduced the window current, and the shift of the steady state activation, again, to the right, in the direction that reduced the overlapping of the two curves. So very, 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 very good question. We never explore in the detail this, but sometimes you hear people saying the calcium channel reactivate. Yes, I don't like that idea, reactivation. these are channels available to open. If you get like a slight increase in membrane potential, open probability increase, this is like a positive feedback. So you approach reversal potential. Let's say you diminish the driving force. Now potassium flow, E-flow, has a good way to pull down. So, and these two forces, right, that are a bit, as I said, you need to think to have an oscillation. You have two forces fighting and a fatigue. One needs to give up. And I guess inactivation of calcium channel may have this role in this oscillation. Yes. I don't think I know. I don't have an answer for that. Oxidative stress exists. I think if it does something similar to what we see with hydrogen peroxide, that it should be like a real happening, in a real cause of arrhythmia in the heart. Hypokalemia also produce this shift. Yes. Yes. Yeah. I think so. I think so. I will show you some data in the full perfused heart and the effect of some drug that have some properties similar to this one, how effective it is. So the good thing is, if you have an early after depolarization, and that is the trigger of the arrhythmia, and we know that cells with EAD tend to synchronize. Right? There is a nice work from UCLA group, like synchronization of early after depolarization, because you understand, if one cell have an EAD at one moment in time and then one slightly shift, there is never enough, depolarizing force to make a bit, a bit propagating. But they synchronize. So you have island where EAD produces PVC, and that's where the extra bit. So, so I believe that these conditions exist in disease heart. Yeah. And so what I show you is really the three most effective maneuver to suppress early after depolarization, right? Again, the reduction of this pedestal, shift of the activation to the right, I think. And I said, these are like really minuscule changes, like a few millivolts we found to be very, very active. Why I like this intervention? And I already mentioned Can you tell me which figure? This one? This one? Yeah. Sorry. This. This is sodium. Yeah. This is the calcium. Oh, this IKR, these are out. Oh, yeah, absolutely. absolutely. You can have this bounce diminishing the repolarizing force. But what the point is, you need calcium, a regenerative current, to propagate. Potassium won't do it. So you need an underlying calcium conductance to make. Say again, sorry. Timing, yeah. But you need a significant calcium current to make the new pulse propagate. Yeah. No, very, very good. Yes. Regionally, yeah. That I said that is the critical. In my opinion, critical to target the calcium conductance. You could achieve something similar, increasing potassium conductance, right? But the effort will be different. And yes, absolutely. No, very good point. So we were here, right? Yeah, and so we took the cell model, and again we look at the L-type current in the model we use for the dynamic clamp, and then we challenge the model, we stimulate the model with an action potential with an EAD, and when we shift, now it's the model, right? We shift the conduction of the calcium current to the right, the midpoint. You see the early peak is not affected. What you see is like more than the late current, right? Peak current is largely in effect, but you have 30% reduction in this late current. You move this curve, the steady-state inactivation curve. Again, this is the green, the blue line. Peak current is still there, but look here, you miss something, yeah? And I guess this is exactly this shaving off of the calcium conductance. It seems so beneficial to prevent the early after the depolarization. And yes, pedestal, the same. You reduce the pedestal, you see the peak current maintained, this is the red line, and then you have less current flowing in the late phase of the action band, especially under the bump of the EAD. Now, as I mentioned before, class 4 antiarrhythmics are very good antiarrhythmics, except that suppress contractility, right? And they do something different. They don't move this curve around. They just push the conductance curve to a lower point, yes? And so the effect is you have both a big reduction in the early influx of calcium, the one that is responsible mainly useful for contraction and easy coupling, and of course also the reduced late current. But the construction is very different in what they are doing. These are calcium channel blocker, and I think calcium channel gating modifier in a way that reduce the late calcium current. This seems to be a better idea for an antiarrhythmic drug. Yes, and then we also tested on the normal action potential, and again, the effect is very minimal on the late current in the normal action potential, while class 4, verapamil, diltiazem, antiarrhythmic of the class 4, they really block peak and late. So now we have this nice picture. So the challenge is, what do? We need to find drugs, ideally, that recapitulate something of this type, which was the difficult part, right? Because, as you can imagine, drugs that selectively do something like this, we didn't know any. So the idea is how to implement these changes. So there is this interesting drug, roscovitin, is used as, I think it's in phase 2 clinical trial, or just complete phase 2 clinical trial, as an anti-cancer, right, is pretty much suppress cell cycle. But it has a nice property, to our point of view. Of course, someone will need to get rid of the cell cycle inhibition, modifying this molecule. But the side effect is an interesting action on the L-type calcium current. You see here, in this early characterization of this drug, roscovitin inhibit the L-type calcium channel. Well, you will see, I don't think that really inhibit. If you want, yes, you can consider it an inhibition, but look what it's doing. This the simple model we have expressed in Xenopus oocyte, all the components of a typical L-type calcium channel. The alpha subunit, the modulatory alpha to delta, and beta to B. Yeah? We add roscovitin, and this is not normalized. Yeah? You have an influx of calcium, the peaks maintain, and then you have these, look like an acceleration of the inactivation. And look at the steady state curve. Activation is not perturbed, but you have this nice reduction in this pedestal current that few years before, in the dynamic lamp experiment, we found to be very effective at suppressing EAD. So we tried. And the effect of this drug is even more interesting in a myocyte. Look, this is an action potential clamp. We clamp the cell with more or less its own action potential, the rabbit ventricular myocyte. Look at the effect of this drug, roscovitin. It's really shaving like the late current, like it was like the dream drug in our view. Well, comparing to a calcium channel blocker, antiarrhythmic, of course, is a big difference, right? Here you understand why there is suppression of contractility. And we think this preserves contractility very much. Maybe, probably we need to do more experiments, but I will show you some data that is very, very encouraging. And let's see a typical experiment of roscovitin. So again, we challenge, we pace a myocyte every 6 seconds. We record high control, action potential. We perfuse hydrogen peroxide. Here it took about, 6 minutes. And then you get the cell completely crazy with multiple oscillation early after depolarization. Actually, you see here, action potential get long, short, because you have some action potential with EAD that get longer, some don't. And then you get in a stable EAD regime. We perfuse roscovitin. Before perfusing roscovitin, all cells have about 250, 300 millisecond APD and 0 early after depolarization. We had hydrogen peroxide. 80% of action potential now show oscillation in EAD. And of course, the action potential gets very much prolonged. Then we add the roscovitin. You see the experiment. You need to restore normal action potential, getting rid of early after depolarization. So this is not dynamic lamp experiment. This is a real cell with his own L-type calcium current, just modified by this effect, I believe, that is this slight change in the steady state, I say, property of the new current. And here is another typical experiment. Here we use a different kind of stressor to shoot. So it's something specific of hydrogen peroxide. So we use hypokalemia, very effective also. We use a combination of hypokalemia and hydrogen peroxide. And again, you induce EAD, and you can suppress with in this case, I think it's 50 micromolar, if I remember, 50 micromolar roscovitin. And see, you go back very much close to, yes? Yes? Yeah. This is the effect of reducing extracellular potassium. Right? You get a change in the, how much is? Yeah, 80, this is about 83 minus 90. Yeah, this is the change in the reversal potential. So here we switch from 5.4 to 2, and so it's very consistent. So we see all the time in rabbit ventricular myocyte. Yeah. Is very consistent. This is also correct for the junction potential. You know, is close to like minus 95, something around there. Many cells. You mean? Yeah. This. Yeah? I don't know. This is very consistent. When we lower extracellular potassium to, usually this is a very good concentration to get early after depolarization, even by themselves, without hydrogen peroxide. And always we have this hyperpolarization. Yeah, I don't know. I never tried, but yeah. Yeah, I can go back and check, but this is like a normal, we so many cells. Always we get this hyperpolarization going from 5.4 to 2K. Yeah, yeah. And then you remember I was mentioning the contractility, right? So fluorophore, we load the cell in control and after Roscovitin to see if there is a change, for example, in the calcium transient that would suggest that we are, affecting the EC coupling or the calcium influx, like you expect that. And as you see here, these are the number of cells we have. There is no really a significant effect on the calcium transient, the amplitude, in the duration, and the cell shortening. So we can take recording of the cell, measure the relative, say, shortening of the cell. And see, with and without Roscovitin, and this is the concentration that usually we use for the suppressing EAD, that there is no much of an effect, which, as I said, this was more or less what we were expecting because of the property of the drug, that really doesn't affect the early peak. And in this experiment, we tested in the, yes. Yeah. Say again, sorry. Yeah. Yes. Very nice experiment. Yeah. So induced by dynamic RAM EAD, and they should be insensitive to. Oh, yeah, beautiful. Yeah. I wish I thought about this experiment. It's beautiful. Yeah, very nice. Yeah. We always use the dynamic clamp to suppress, but it's a great idea. Yeah, that should be absolutely insensitive to Roscovitin unless there is another mechanism. Yeah, beautiful. Yeah, no, thank you. The effect of Roscovitin in an artificial current. Right. No, nice. More? The experiment of the this one. Yeah.
So what is you say that's the control, but that's the control in the present. Current there, the pedestal is
yes.
In the presence of hydrogen peroxide.
Yes, this is a control independent of hydrogen peroxide plus roscopic. Yeah, this.
So what does the real control look like for drug just?
No, sorry. This is not. No. This is not. Sorry, no. This is not. These are normal myocytes. Excuse me, oocytes in expressing the 3 subunits before and after roscovity. Yeah, no, there is no hydroperoxide. No, this is the normal cell. Sorry, yeah. That to be a myocyte, yes. Well, it's a bit different. This is an oocyte. Yeah. Exactly. Yeah. Yes. Yeah, the kinetics in the myocyte is faster. Yeah, this is like the best just to test the drug on a simplified, like 3 protein system. Yes. And I said, these are a series of experiments that we did in collaboration with Cargusian lab at UCLA. So they use age rat. Age rat are, it seems very easy to induce arrhythmia by oxidative stress. You see, you need lower concentration than the one we use in a single cell. We need to go up to 600 micromolar hydrogen peroxide. But with 100 micromolar, really, you see, in 38 minutes, this is a perfused heart. In a Langendorf, we deliver hydrogen peroxide. You can have the heart going into arrhythmia, or excuse me, VF. Then we add roscovitin, and in 16 minutes in this experiment, and 20 micromolar roscovitin, the arrhythmia terminates. And see, so far we have, 8 heart, and all 8 rabbit heart, excuse me, rat heart, in an average, in 13 minutes, roscovitin terminates the arrhythmia. Now, if you don't stop the arrhythmia, when we induce this, the heart dies. It becomes asystolic after minutes. So the only way to keep the heart alive is to terminate VF, or do I keep going? And we found that roscovitin was actually terminating the fibrillation there. So where this fibrillation was coming from? Of course, it has to be like a focal reactivation of a part. You know, for the expert in the arrhythmia field, if you have like a mother rotor or something sustaining the arrhythmia, I don't think shaving the calcium current could help. But we have a focal reactivation generated by PVC or EAD, then this intervention should work. And apparently this is what is happening. This is another thing. I said hypokalemia is a very good way to induce VT and VF in animal, again in rats, heart, perfused under Langendorf. And again, here, after inducing VF, you see these are again bipolar electrogram, one placed in the left atrium, right ventricle, the pseudo ECG, which is pretty much the difference between this and this, more or less, and a microelectrode there to sample different area of the myocardium. In 8 minutes, 20 micromolar scopotin again terminated the VF. And in this case, again, it worked in 5 out of 6 hearts that we have, perfused with roscovitin. And we also tried some preventive experiment in the rabbit heart. So we were actually perfusing roscovitin together with this extreme hypokalemia, you see, 1 millimolar plus 100 micromolar of hydrogen peroxide. And pre-treated briefly for 15 minutes. Sometimes we are at the same time with roscovitin. And you see, this is like a survival curve, survival of the heart, let's say. You see heart without ventricular tachycardia or fibrillation. Without roscovitin, and we had 5 animal rabbit heart for each condition. Without roscovitin, yes, you lose all of them within 40 minutes in average after arrhythmia. With 20 micromolar roscovitin, you start seeing some improvement. And with 50 micromolar roscovitin, we only lost 1 of the heart in 1 hour out of 5. And this is a prevention. Like, yes, we have 15 minutes of roscovitin and then this is. So you ask me why this hypokalemia? Well, because it's very difficult to induce VTVF in young rabbits. And we found that this was the best combination, 1 millimolar hypo-K and 100 micromolar angioperoxide peroxide. Yeah. Age rabbit are, for some reason, very hard to find. We need to get one once in a while from a company that use mainly to produce antibodies. Yeah, you're right. Re-entry, but sustained by new event, like that what I think. But why I could not explain why it's suppressed. Like this, like curing VF. So it has to be new, like a focal reactivation that sustain the arrhythmia. That what yeah. These are all very good points that we are still, thinking about. Yes, this is like a very entral wave. I never test this drug or this idea. What is the effect of roscovitin on a meandering wave? Yes, that probably we should do if. We started with some monolayer in which we can actually observe the mapping. No. But it will be very, very, very useful. Very, very, very useful. Yeah. No. Yeah. No, it will be fantastic. If I find someone that want to map this, this one. These are, yes, we start this experiment like a yes or not question. We induce arrhythmia, so we break the normal pattern. What happened with this drug? Like the mechanism is very interesting, and we didn't really work it yet. Sodium, this compound has been studied. I remember, for sure, is blocking, in part, IKR, which may be a beneficial thing. Like if you block IKR, you should be APD prolongation, right? And we don't see it. I don't know if the concentration of the system is not sensitive. The channel are not sensitive enough to roscovitin, so we are in the good window of concentration, or there is like maybe a slightly compensative effect. So blocking YKR tend to prolong, but since we are shaving a bit the calcium influx, tend to reduce the APD. See, I don't remember this, but I'm sure someone tried. So the fact that I don't remember to have seen a sodium channel effect of this, maybe they didn't find it. Why you are asking this? Like a mechanism to stop the... Yeah, no, it's a good point. Right. No, as far as I know, it's not blocking sodium carrying. Yes, we should probably also try in this system, rabbit, and yeah. What is known about them? Yeah, the mechanism is unknown. How do you think? There are some work where they have, from chimeras work, so the effect of roscovitin is something that has to do with domain 1 of L-type calcium channel. So when you replace domain 1 of the L-type, become sensitive to extracellular roscovitin. How is producing that effect? I don't think that has been worked out, that one. And but again, it's a very good question, but maybe not important for... So yeah, let me clarify one thing. I'm not saying, and I don't want absolutely to propose that roscovitin is an antiarrhythmic, right? Roscovitin is an anti-cancer. The side effect, unwanted effect of this calcium, I think it's very intriguing to me that if we could design drug with this property, they could work as a good asset. So I see roscovitin as a prototype, an idea of what a drug should do, right? Regardless of the mechanism. I mean, like if you can reduce this late current and change, in the case of roscovitin, this like the pedestal non-inactivating component of the late current, to me that would be a very powerful tools that could, yeah. So sodium hydrogen peroxide at the end activates CAM kinase 2, and then the phosphorylation of everything, sodium, potassium. So the overall effect on the cell is probably pretty bad. Still, that's the point. When you had this drug, or in dynamic clamp, you correct this, the property of the L current is very effective. So that's why, when we started this project, I never thought I was going to test the roscovitin or some drug with this idea. We were very happy. I'm an ion channel biophysicist, where I found a nice correlation between EAD and the position of this steady-state curve. So, but that, the project evolved, so I said, why don't we actually look at how this thing works? So that's more or less the history of that. And, so I'm very late, right? Yes. So, there's another idea. When we had gabapentinoid, pregabalin, to the myocyte, we had noticed this shift of the activation curve, right? And again, this is one of the maneuvers that we think it should be effective. And you see, it looked like a real shift, because the maximum conductance, at least in this cell that we are exploring, we couldn't see any blockade of the block of the channel. If you depolarize strong enough, you open them. And this is a ton of pregabalin, yes. These are gabapentinoid, they bind to the alpha to delta subunit of calcium channel. So alpha to delta subunit have a strong modulatory effect on the L-type channel, and the idea was, well, maybe pregabalin binding there attenuate a bit the effect of the subunit, letting the curve moving few millivolts to the right. And again, this is you are very familiar with this type of experiment. We challenge the myocyte with hydrogen peroxide, and then with pregabalin, we could stop EAD. This is moving, I think, this effect has shift the voltage-dependent activation. Right, remember, this is the second maneuver we found very effective. And you see, in control, you add hydrogen peroxide, you get all cell with EAD, and you can return them to normal baseline with pregabalin. It's a bit high concentration. I recognize it's the best drug I can find. Actually, if you can tell me, any drug able to change the voltage dependency of a calcium channel in that direction would be great to test. And again, it was effective at suppressing in the same system. This age rat suppressed 50 micromolar, suppressed VF induced by hydrogen peroxide, in this case within 8 minutes. And in total, we have 5 animals now that respond to this intervention. And so I want to conclude here. I just got this simulation from Zilin Chu, many of you probably know, at UCLA, and his student Mike Chu. I didn't have any idea of how to test the left shift of the steady state activation. I don't know any drug doing that, so I couldn't test it. So I asked them, why don't you simulate in your anatomic heart simulation? These are like a 2 million cell. These are only the ventricle. I got this yesterday night in my hotel, and it's just probably very premature, but I love to show it. How do I start it? Maybe I need to do here. And I conclude. Yeah, I cannot even start it. Sorry, it's fine. But in this experiment, they have, so this is a local O'Hara-Rudy model for LQT. There is an enhanced calcium current. I think they double the L-type calcium current, and they remove IK1 completely. So the model breaks in fibrillation after a few minutes. They have shifted the steady state inactivation curve of the L-type calcium current by 5 millivolts. And if I could show you the simulation, the model doesn't break anymore. So this is as far I can go with the third maneuver. Like a simulation is, I don't know any drug doing that, but it would be fantastic to try if you have any suggestion. So yeah? Oh, yes, it was going? You saw it? I don't know. I'm very really. I put it in my presentation yesterday night. I was already sleeping. I didn't test it, unfortunately, today. My apologies. But what I want to show is, is it so? Yeah, it's going to the, I don't know. It's beautiful, huh? I need to tell you. We are missing something very nice, but it's okay. I don't want to take your time anymore. So what I would like to convey, in summary, is that, what? So blocking calcium channel is a good idea as an antiarrhythmic. That's what I'm convinced, and what cardiologists at UCLA convinced me. I don't know if they are right. So, but the good news is we don't need to block them. We just need to slightly change few steady state property, very simple, of the L-type calcium channel to suppress early after depolarization and EAD-mediated arrhythmia. So, and I think that could likely be a new class of antiarrhythmic drugs, no? That very differentiate from the class 4 that we have now. And so this is what class 4 antiarrhythmic are doing. Very nice antiarrhythmic, but with negative inotropic effect. They remove early after depolarization. They block calcium influx. Yeah? These are two maneuvers that I show experimentally using two drugs that the best I could find to approach this idea of moving this curve. I show something about roscovidine and gabapentinoid. Yeah, they are very effective at abolishing early after depolarization. They block arrhythmia, or at least they rescue VT and restore sinus rhythm. And for the third one, I didn't have any drug, but if I could show you the simulation, it would be a nice thing. It didn't work, but the good news is, at least in the model of a full heart, even the third maneuver seems effective at suppressing AD. So I want to conclude here and thank you for your time and attention. And there are incredibly dedicated people that have worked. This is work going on for many years. Marina Angelini, Nicoletta Savalli in the lab, Federica Stecanella, Rojni, that did the PhD in the lab, starting with the dynamic clamp experiment, which we learned together actually had to use and perform dynamic clamp experiment. Antonio Pantazis, that just got his faculty position. He moved to Sweden. He's an associate professor now. And Araj, all the experiment with the perfused heart. And Mike Lu, he did a PhD student of Zilin Chu at UCLA. He's the one that sent me the beautiful, you need to believe me, whole heart simulation that I cannot play, unfortunately. And a fantastic group of colleagues at UCLA, Jim Weiss, the chief of cardiology, Haraya Karaguzian, Zilin, Alan Garfinkel, and other people that have worked in different aspects of, especially the roscovidine project, that we are still trying to learn what is doing. And I thank you very much again for the invitation and for your attention. Yeah, these are all are affecting voltage-dependent activation. All these maneuvers.
Yes, I wonder if you've looked at.
Yeah, this is a very good question. So to have a selective intervention to change calcium-dependent inactivation. Yeah, probably I think it would work equally well. Yeah, we can do that. But the thing is that you are absolutely right. There is more to explore. I mean, the dynamic clamp, when I realized the power of this technique, really, I fell in love with dynamic clamp. You can explore a number of conditions and an idea in a reasonable time. And yeah, so it's just, I think I'm limited only by my imagination what I could do with that system. We transition to the point to say, okay, can we find some drug that actually recapitulates some of these things? And this is the best I could find to, prove or disprove these ideas. And but yeah, if you have any idea of any drug moving this curve without affecting too much of all the conduct, that would be fantastic. Yeah. As I said, alternative stimulation should, push the curve in the direction that we see like dangerous or So you asked me like to do an experiment with isoproterenol, for example, and test this maneuver. Yes, absolutely. But again, the curve will shift, the activation curve will shift in the direction like hydrogen peroxide is doing. So it should work. It should absolutely work equally well. A drug like, that's the thing. I think what we is coming out from this study that, yes, the insult is moving all these 3 parameters away, but you need only to correct one to not to restore, to diminish the probability of oscillation, which is what the whole idea is about. Is about diminishing the probability that you get that oscillation going. And you have a very good point. We should try different stressor. No. Yeah. You me? T-type. We don't have. No. Yeah. Yes. You are right. It could rescue some steady state inactivated T-type. Yeah. Very good point. Yes. No. It would be good. Yeah. Yes, absolutely. Now I need to go back and check if the resting potential drops. No, but this is like one of the few things that we always observe. But you think it's too much? Yeah. There is 13 millivolt. Yeah. Yeah. I need to say, my experience with cardiac myocyte is all about rabbit ventricular myocyte. So this is the only system I ever work with. So, no. Yeah. Okay. Say again, sorry. Yes. Roscovitin. You are talking about Roscovitin? Yeah. So I took Roscovitin straight out from the war of drugs because so it's safe. It's already in phase 2 clinical trial, so it has no adverse effect. So, and I read a bit, there is no cardiovascular effect reported in this clinical trial. So, I don't know if that Roscovitin will affect normal, healthy patient. The only thing I have is that simulation. Now I don't want to bother you again, going back the thing. But you remember, in the normal axial potential, without EAD, when at least we simulate the effect of roscovidin, diminishing the pedestal, there was almost no effect on the normal calcium current. You remember, I don't want to work, but practically it was untouched. Yes, when you have an EAD, yes, the peak was maintained, but then the intervention reducing the pedestal was also taking out current from the late phase, which is what we think is the beneficial aspect of the whole maneuver. Oh, yes, you're right. Like our, let's say, baseline, is true. Yeah, then I don't know, maybe we should maybe let roscovidin there for 1 hour, yes, and see more long-term effect. Absolutely, yeah, that's a good point. Yeah, thank you.